Hi there, my name is Daniel Petroscu. Today I am going to be doing a presentation over the research paper Reversing a Model of Parkinson's Disease with In Situ Converted Nigral Neurons. This paper was done by Kien et al. This paper was done in order to see how conversion of astrocytes into neurons could affect Parkinson's disease models of mice. To bring a little bit of importance to this topic, we are going to start off by talking about Parkinson's disease. Parkinson's disease is a very important disease as it affects a multitude of different people in America and throughout the world. Every 6 minutes, someone is diagnosed with Parkinson's disease. And what we see is that there's a higher amount in males. It gets worse with age. So most of the time, people above the age of 65 will be affected by Parkinson's disease. It also has multiple different symptoms involved with it. So a lot of it is loss of motor control, such as tremors, rigidity, slowness of movement. It also inhibits motor learning, and it inhibits some mood as well, causing some things like anxiety and depression. What this disease is really caused by is the loss of dopamine in the substantia nigra, and this causes a bunch of different circuitry to be messed up, to degrade, and causes brain degradation throughout the process. Our current treatments are not the best kind of treatments right now. We have L-DOPA, which basically increases the amount of dopamine in your brain, and you have deep brain stimulation, which basically activates some parts of your brain. Again, these are not the greatest treatments, mostly because they only really help with motor symptoms. The problem with these treatments is that they do not actually stop the degradation of the disease. And even with motor symptoms, at some point in the disease, the effect that these have as a therapeutic device do not work and they do not help retrieve motor symptoms. So why is this happening in the first place? And a lot of it is because of the neurons that are involved in this. Most of these neurons are found in the central nervous system, and these neurons are your dopaminergic cell bodies. In Parkinson's disease, these neurons are destroyed, which causes neuronal circuits to be degraded, causing decrease of dopaminergic cell bodies. The problem is that these neurons in the central nervous system, unlike your peripheral nervous system, cannot proliferate. So there's not really a way we can regenerate our neurons in Parkinson's disease patients, even though degradation is occurring. Astrocytes make this a little bit more interesting. So we've talked about astrocytes in class a couple of times. Of course, they're important for your tripartite synapse. They're the most abundant glial cell in the brain. They also provide nutrients to the blood-brain barrier, but the most important thing that astrocytes provide is that they can proliferate. This is really important when we talk about the importance of this paper in specific. This ability to proliferate builds up the question, can astrocytes be used to rebuild circuitry? If the astrocytes can proliferate at high capacity in comparison to neurons which can't proliferate whatsoever, could we use this ability to kind of rebuild the circuitry that we see in the brain degradation of Parkinson's disease? Again, et al. Look to answer this question through their paper, Reversing a Model of Parkinson's Disease with In Situ Converted Nigral Neurons. So the basis of this research begins with our fibroblasts. Fibroblasts can be converted to neurons through a RNA protein called PTB. PTB is mostly used to control neuronal maturation. And what we can see is that when you deplete PTB, there is a conversion of fibroblasts into neurons. So I have this little figure up here. I'm going to go more in depth about it on the next slide when we talk about the astrocytes and neurons that also have this similar pathway. Astrocytes have the exact same pathway, but I'm going to go over it in the next slide. And another basis for research is that glial cells have high ability to proliferate, especially astrocytes. So this can be used as a way to regenerate neurons. To talk about this pathway, I'm going to use a little like analogy. But to start off, I just want to introduce all the ideas. So there's three important neuronal genes that will activate the ability to turn into a neuron. Well, there's two proteins or slash neuronal genes that will inhibit that ability. So the MIR124, the MIR9, and the BRN2 are all important in the maturation of neurons. Your REST and your NPTB are important in inhibiting neuronal genes. So how does this even occur in the first place? Well, in a normal neuron, there is no PTB. In a normal neuron, MIR124 will block rest and will block PTB. NPTB, which is induced by PTB, will then block BRN2 and MIR9 eventually. But because there's no NPTB or PTB, this doesn't occur, and BRN2 and MIR9 can be used as normally. When PTB is induced, we see that it blocks the MIR124 pathway from blocking rest, causing a different negative feedback loop in which rest will block MIR124. PTB also induces NPTB, which blocks BRN2, which will block MIR9 because there's not expression going from BRN2 to MIR9. To make it a little bit easier to understand, just because I thought it was a little bit fun over here, I use this example of a security guard. So we have this little security guard over here. He's a little silly guy, but basically he will be your PTB. Let's say there's a cell, not a fully developed cell, a cell that wants to become a neuron. If he gets past the PTB, he will go into this box right here. This box will allow him to express MIR124, MIR9, and BRN2. This will allow conversion into a neuron. However, if one of these cells goes and meets up with PTB, then we will see that he will be thrown into this box, which will force him to express REST and NPTB, which will cause conversion into an astrocyte. Now we're going to talk about how this is even possible. How can we convert the astrocyte into a neuron by removing this PTB? The way researchers did it in this paper is by introducing an AAV. An AAV is an adeno-associated virus. Basically, it's just a vector that has some genetic information. And more importantly, is that they have this little short hair RNA loop, which you can see right here, for PTB. This loop right here, alongside the adeno-associated virus, will be injected into the brain, which will cause removal of this specific protein. This, in theory, should cause conversion of astrocytes into neurons. To test this, researchers did a little bit of staining. They used RFP, which is one of your fluorescence markers over here, and they tested for NEUN, which proves that a cell is a neuron. What you can see is that at first, 3 weeks after being injected with the AAV and the short hair RNA, you saw that there wasn't too much expression, but as the weeks go by, you start to see a lot of collateral expression. Important thing to note is that this was done in the midbrain. We'll talk about the reason that's important, but you will see that there's some regional specificity to this when we talk about dopamine being released. Another thing that is important to know is that alongside increasing the NEUN, which conversion into neurons, we saw, especially in the midbrain, that this adeno-associated virus with the short hair RNA caused for an increase in DDC and TH. These two enzymes we've talked about these two enzymes in class, in which they turn tyrosine into dopamine using tyrosine hydroxylase, which is your TH, and your dopa decarboxylase, which is your DDC. And you see that there's an increase showing that there's an increase in dopaminergic cell bodies. And then in here, just to show talking about regional specificity, what we can see is that we have NEUN and the RFP, the marker. Researchers expressed it in the midbrain, the striatum and cortex, and saw pretty equal amounts throughout, showing that astrocyte conversion into neurons can occur at any place in the brain. However, after the conversion from astrocytes to neurons, they also tested for tyrosine hydroxylase, and they saw a lot more overlap in the midbrain rather than the striatum and cortex, which is also shown by this graph. So this specific conversion has regional specificity when talking about dopaminergic neurons. Let's talk about our mouse model that the researchers are going to use. They will be using the 6-OHDA model. This model can be used on either hemisphere of either side of the brain, in which researchers inject 6-hydroxydopamine to one hemisphere, one side of the brain. This could be located in the striatum or the substantia nigra. Basically, the idea is that it will result in a loss of dopaminergic cell bodies in one specific area. And what researchers did in this paper is that they cause the lesion in one side of the brain while leaving the other side of the brain intact so that they can compare the two results between the two hemispheres. What this also does is it causes loss of motor symptoms similar to Parkinson's patients. Something that's important to note is that this is not a perfect model. We'll go more into that when I start to talk about the limitations of this paper. And just to give a little brief idea of like the timeline that occurred throughout this through this research, when before 1 month before they injected the adeno-associated virus with the short hair RNA, they had the 6-OHDA lesion occur. 1 month after that, they also tested to see the cell body counts in the 6-OHDA lesion. You'll see that in the next slide. As a month after the lesion, they injected the mouse with either a normal adenosine-associated virus with no vector, with no short hair RNA, versus one with short hair RNA for your PTB. They also did a behavior test. Then 2 months in, they did a normal behavior test. 3 months in, a behavior test and quantifying the amount of cell bodies. And 5 months in, a behavior test and quantify the nerve fibers that are formed. So to show what I was talking about with this model staining or this count of normal neuronal cell bodies, researchers use a couple of different markers here. They use TH, tyrosine hydroxylase, DAPI, which is a fluorescence marker, RFP, which is a fluorescence marker, and GFAP, which is a marker for glial cells, most importantly, astrocytes. What we can see is that 1 month, this staining was done 1 month after 6-OHDA lesion. There was no expression of dopaminergic cell bodies 1 month after lesioning occurred, meaning that 6-OHDA worked and caused decrease in the amount of cell bodies. What researchers also saw is that the intact side of the brain had a good amount of expression for TH, showing that they were producing dopamine, while the lesion side only showed GFAP, so mostly astrocytes. What's even more interesting is that when you add the short hair RNA to the adenosine-associated virus, we saw that in the intact substantia nigra, there was a good amount of dopamine being released. In the lesion side with no short hair RNA, there wasn't any release. But when you added the short hair RNA, there was astrocytes, of course, but there was also a good amount of not astrocytes, just marking. There was a good amount of overlap with tyrosine hydroxylase and the expression in that section of the brain. They then also compared how the adeno-associated virus without a short hair RNA and with a short hair RNA. This was about 10 to 12 weeks after the original AAV was added. What you could see is that this would be the intact side that they had. The intact side, of course, normal amount of cell bodies. The empty side without the AAV had no cell bodies, while the short hair RNA had a decent amount of cell bodies, not as much as the intact side, but more than the non-intact side without the short hair RNA. They then did a close-up to do a little bit more staining and found that there was a pretty overlap of tyrosine hydroxylase with the fluorescence marker, showing that dopamine was being produced by these cell bodies. So to further drive that point across, what you can see here is that you have an intact substantia nigra on both sides here. You see that there's a good amount of total cell body that are there. After you have a lesioning occur without a short hair RNA, you saw that there was a major decrease in the amount of cells that are in that part of the brain. However, when you did add the short hair RNA in this section right here, you saw that there was an increase or a rescue in the number of total number of cell bodies. We also saw the same thing with the relative tyrosine hydroxylase fibers. You had a normal amount of cells, or a normal amount of percent density, a massive decrease around to 10%, and then you had some relative amount of rescuing that occurred. Well, we also saw the same thing in the amount of dopamine found in the striatal dopamine. So on the intact side, you had 100% of dopamine being released in comparison to, of course, the intact number of dopamine being released. When you had the adeno-associated virus without the short hair RNA, you saw that levels of dopamine decreased down to about 20%. And then when you added the short hair RNA alongside the adenosine-associated virus in the 6-OHDA lesion, you saw a return to about 65%. So there is some rescuing of dopaminergic cell bodies in the amount of dopamine being released. Where researchers went from here is that they looked at the amount of the motor systems and how there was rescuing of the motor symptoms. The way they did this was through a drug-induced rotation test. The drugs they used were amphetamine or apomorphine, and basically the researchers injected the drug into the mice, and the mice would move in a certain direction. If there was no lesioning occurring, then the mice wouldn't move at all, showing a normal amount of motor control. However, if lesioning did occur, then you would see a rotation in that specific direction or a loss of motor control. For amphetamine, it was an ipsilateral movement. I think ipsilateral was defined as left, while on the other hand, the apomorphine went contralateral, which was defined as right. Before the adenosine-associated virus occurred, you saw that there was a pretty like similar amount of ipsilateral and contralateral movement occurring. But 3 months after the adeno-associated virus was added, we saw that when there was no short hair RNA, there wasn't really any retrieval of motor symptoms, while when there was a short hair RNA, there was complete retrieval of symptoms. This was shown for both amphetamine and the apomorphine, showing that when mice had the short hair RNA, they were able to fully rescue their motor movements. And we saw kind of the same thing as well, just as another way that they graphed it. However, this is showing the time. So what we can see is that for our lesion plus our AAV minus our short hair RNA, there wasn't any retrieval of motor symptom or rescuing of motor symptoms. On the other hand, when short hair RNA did occur, as time went on, we saw that there was an increase back to baseline of motor control. This is important because it shows that that researchers have a way to kind of fight against the Parkinson's disease degradation. The short hair RNA seems to be making somewhat of an impact over time, which can help combat that. So overall, the reconstruction of the circuit can work. I say can because we saw that motor symptoms were fully rescued, but the dopamine levels were recovered only to a little bit of an extent. What's more interesting is that this conversion took place, the astrocyte conversion took place over time, meaning that as time passed on, these results only got better and better. So this kind of shows a way in which researchers can fight against the degradation of the brain. However, there were a few limitations that did take place during this time. So, of course, it's not completely safe to inject humans with an adenosine-associated virus that was made for mice. Mice express different proteins, different genes. So, researchers need to find a way in which they can make this safer for humans to have before this can be rolled out. Researchers also tested for age in conjunction with disease. They tested it a little bit, but not enough to have big results. One important thing to note was they tested mice that were older for motor symptoms with the adenosine-associated virus and the short hair RNA. And what researchers found was that there was no improvement in the motor symptoms. So this could be used for future research to see if there is a way to counteract this. And of course, the 6-OHD model is not perfect. While it does induce some Parkinson's-like symptoms, it does not completely replicate the effect of Parkinson's. And this is for a couple of reasons. The 6-OHDM model is just a quick burst of a quick lesion, while the Parkinson's disease is a long time degradation that can happen over a couple of years. Also, the 6-OHDA model only affects like one specific region of the brain, while Parkinson's disease, if you see a brain of Parkinson's disease versus a normal brain, you see that Parkinson's disease brain has a lot more degradation in also the frontal cortex and other parts of the brain rather than just the substantia nigra. So this is also something that needs to be kept in mind. There isn't really that great of a model for Parkinson's disease. 6-OHDA is one of the only ones. There's also MPTP, I believe, but these are not perfect and they do not completely replicate the results. As for future research and where researchers can go from here, could we use astrocytes? Could astrocyte conversion be used in other circuits and diseases? So maybe this conversion could be important for Alzheimer's or dementia or Huntington's disease. Could other areas of the brain outside the substantia nigra also impact these findings, which might be important? Of course, you know that the substantia nigra is mainly producing the dopamine, but maybe it could be used for maybe other areas of the brain could also be helpful. And like we also use other glial cells for conversion as well. Maybe microglia can provide a different result. So these are things to keep in forward as researchers continue on with this disease. These were the references that I used for my research presentation. If you have any questions, please just let me know, and have a great day.